Introduction to the reference view. VGAS requires all sequence datasets to be aligned to a reference. The example we're using here is HRV1 and the HXB2 reference strain. All feature types, i.e. genes and coding sequences, etc., are separated into 3 groups and displayed in these panels. The genes are represented by the blue bars within the gene panel. The panel above, displaying the yellow bars, is called the feature focus panel. Any type of feature, apart from genes, can be selected and represented as yellow bars. At present, the coding sequences, or products, are selected as the feature focus. The coding sequences are always selected by default. The panel containing the smaller grey bars is called the Miscellaneous feature panel and shows all the other features. These can be coloured by feature type. When a feature type is selected to be the feature focus, it's swapped around from the Miscellaneous panel to the feature focus panel. The track bar to the right allows us to zoom in and out of the features. If we zoom into 100%, then one nucleotide base is represented by 1 pixel on the screen. At the top, the blue bar represents the length of the reference genome map, and the red bar indicates the region of the genome that is currently visible on the screen. Therefore, if the entire reference is visible, then the bar is completely red. All these panels can be hidden from the View menu. At the bottom, we have the reference sequence, with the scales showing the reference nucleotide base position. The bases on the forward strand are shown at the top, with the corresponding complement strand below with the bases slightly faded. We also have the 3 reading frames of the amino acids. Each residue is centered to the codon, i.e. in the middle of the 3 nucleotide bases. A single mouse click on either gene or feature focus highlights the feature in pink. A double click selects the feature and colours it in green with the sequence shown below. Another blue bar now appears at the top, which represents the feature selected, with the red indicated the region of coverage of the reference sequence showing. If we right click with the mouse on the reference sequence, a menu appears. This lets us select the type of scale to be shown. Either the genome nucleotide base positions, the feature nucleotide base positions, or the feature amino acid residue positions. We can also copy the reference sequence to the clipboard in various formats. The current nucleotide base position will always be in the center of the screen, highlighted by the center bar and the blue line through the panels. Therefore, as the screen is resized, the display will always remain centered. We can also right-click on any of the feature bars, which will present us with various options. One being to export the image to a various image type. We can generate an image in various formats for the full reference genome or for a required region. The scale of the image will match the current scale displayed, which can be altered by the trackbar. Clicking on the double arrow button on the top right will display the custom location order grid. Here we can create one or more custom locations on either the forward or complement strand. To remove the custom location, unclick the button. The previously entered locations are also stored. Clicking the button on the top right opens the Reference Genome grid. The grid lists all genes and feature types associated with the reference sequence. These can be ordered by clicking the arrows in the column header. Selecting any gene, feature or feature location will move you to and highlight these in the reference view. From here, we can also select the feature to be displayed in the feature focus panel. Currently, coding sequence is selected. If we select another feature, say mature peptides, then these are now displayed in the feature focus panel as yellow bars. So therefore we can now select any of the mature peptides and the reference sequence will be shown. We can also change the colour of any miscellaneous feature. And the information panel gives a description of these. Highlighting any feature from the grid and right-clicking the mouse displays an option menu. Here we can copy the sequences into the clipboard and paste them in FASTA format. Selecting copy location orders from the menu allows us to paste these into the custom location order grid. Clear, paste, set locations. So you can see we selected GAG, TAT and VPR. This example shows the EBV reference sequence. Here we notice that some of the genes and feature focus are displayed as a slightly darker blue or yellow bar. These are features which are located on the complement strand and scroll backwards along the genome when selected. The scale also shows this. EBV also contains genes and features which are spliced over the entire genome. If we select the gene display option and show location order, then we can see the number of splices that the feature is made up of. This feature has 11 splices spread over the full genome. The splices are separated on the reference sequence by a red dotted line. So you can see here, and we're at the end of the 8th out of 11. If we then jump to the next one, we jump up to the 9th out of 11. If you choose to copy the DNA sequence, then the sequence is actually spliced together.